And here we go, on to a discussion of DNA and its role in determining our traits and also in genetic disease. We've looked at DNA somewhat already in class and considered that DNA is a nucleic acid. It's one of two. RNA, of course, is the other. And we talked a little bit about what DNA does just in reference to it's involved in our heredity or it's involved in our traits. What do we mean by that? What does DNA actually do? What's its role in the body? And how does DNA direct protein synthesis? And how do genetic mistakes result in disease? When you think about DNA's role, it is central to protein synthesis. Here we have in the nucleus, we have DNA. It's partially unwound here so that we can see it a little easier. But DNA in the nucleus is going to be coding for or responsible for ultimately the directions that make our proteins. By that I mean consider DNA is serving as a blueprint. Coded within the sequence of A's, T's, C's, and G's is an order that dictates the order of amino acids, which you see down here. Aspartic acid, arginine, and the ones that are already on this chain are amino acids. How did this order actually occur? Well, the directions, again, are in the sequence of A's, T's, C's, and G's in the DNA in the nucleus. I'm not that concerned that you are familiar with the details of transcription and translation, which are the two main steps for how DNA becomes encoded, or the DNA code will be used to produce the amino acid sequence. But just to briefly show you what's going on here, in the DNA, as DNA is unwound, RNA is going to be copied from it in a complementary fashion. And again, the details really aren't that important for today. But an RNA copy will then make its way out of the nucleus and into the cytoplasm. Once it's in the cytoplasm, the RNA is going to be read. So this piece right here, the messenger RNA strand, is the same one that we saw here in the nucleus. It's just blown up for us to see a little better. So if you look carefully, you see A and G and C and U and A and G. We have the code that came from the nucleus, which came from the DNA itself, is now used to put together the order of amino acids. And again, you may be familiar with how this is done. If I have a CGA that corresponds to a GCU on a transfer RNA, and if those details are a little fuzzy for you, don't sweat it. Just understand that this order of messenger RNA bases dictates the order ultimately of the order of the amino acids. Here you see arginine is the next one locked in place. Ultimately, I'm going to get a polypeptide chain which will fold into a functional protein. But understand, where did that order ultimately come from? It came from the DNA sequence, the sequence of DNA that you inherited from your parents. So this gives you just a little bit of an overview of the processes. Again, here you notice that we have DNA, RNA, and protein, and ultimately the protein will result in a trait. Let's say the protein being made here is coloration for eye color or hair color. Once again, you see our steps, transcription and translation, just showing you the process. But do recognize here, the trait ultimately goes back to the DNA sequence that you had in the nucleus that will then be read so that we can produce things like structural proteins, which result in traits, enzymatic proteins, which result in metabolic traits, whether or not, for example, you are lactose intolerant. Depends upon the enzymes that you can make. The enzymes you can make depend upon the DNA in your cells. Hormonal proteins like HGH may be, they are the result of DNA sequences that ultimately control our growth and development. So a gene is a segment of DNA that is going to be coding for or is responsible for ultimately the production of a particular protein, which will then result in a trait. Variation in this, though, can result in problems. What you have here, we're seeing the normal sequence of a GCA, AGA, GAT. Again, the sequence of bases in DNA, in this case, spells out the amino acid alanine, the amino acid arginine, the amino acid asparagine, and so forth. And ultimately, we end up with a functional protein. The order of those amino acids results in the functional protein. You could have a change in the DNA, like you see here. We've changed an A from here to a G, and notice just because of the way the DNA is coded from DNA to amino acids, that particular change didn't cause any change in the amino acids, so there was no change in the protein. Sometimes a change in the DNA doesn't matter in terms of changing the protein shape. However, in this case down here, you see that it does. We changed this G here to an A. Well, that resulted in an entirely different amino acid, so we get an entirely different sequence of amino acids, which will then cause maybe the protein to fold up in a different way. That could be a genetic problem. Let's say it's something like lactose intolerance. The correct version of lactase may have been this shape. The incorrect version may have been that shape. And because it's in that shape, from what you know about enzymes, shape means everything, and if that's the case, that protein, that enzyme won't work. So what can cause these variations or these changes? Sometimes it's just that when DNA is copied. Sometimes environmental agents like radiation or chemicals can change DNA, or sometimes other factors can cause misshapen proteins, and we'll talk about those a little bit later. Let me give you just a couple of examples to close this video. Albinism, people who are albinos, that's one example of one of these metabolic disorders. Notice in this case we have our sequence of a biochemical sequence from phenylalanine all the way ultimately to the production of eumelanin and this red pigment form of melanin you see here. If we have all of these enzymes, you see the ASEs, if we have all of these enzymes and everything goes according to plan, that is, according to these metabolic pathways, we produce the black and red pigments that we associate with melanin, and someone is of quote-unquote normal coloration. However, if there's a genetic disorder where, for example, this enzyme is not functional, if this is not functional, then all these other pathways don't happen, the melanin is not produced, and someone has albino characteristics. Let me just show you one more example. Tay-Sachs disease is a progressive neurologic disorder. Babies will begin to develop the symptoms usually around 6 months if they have this condition. In this case, they have a defective gene. It's called a hexagene. This hexagene is going to be involved in controlling lysosome activity. Particularly, there's an enzyme that controls the way we break down products in our lysosomes, and we'll discuss that more with cells. But if that is not functional, then there becomes progressive nerve cell damage, and eventually we end up with damage that we associate with Tay-Sachs disease, and these babies rarely live past 4 years. So genes are important. Our genetics, our DNA is important for reasons you see here, that DNA does control traits, and defective DNA can result in disease.